Hi, everyone. Thanks for joining us. My name is Amy Franco, and I'm the Assistant Director of the Adult Department at the Glen Ellyn Public Library. This program is a joint partnership of the Glen Ellyn Public Library and the Orland Park Public Library. Welcome to our COVID Q&A, with our special guest, biomedical engineering researcher Rebecca Keate. Rebecca is a PhD student at Northwestern University and is generously volunteering her time and expertise to answer questions you have about COVID-19. Rebecca researches electrically conductive biomaterials for tissue engineering and holds a BS in biomedical engineering from Georgia Institute of Technology. Her research includes bacterial infection, cellular mechanisms of cancer, and tissue mechanics. I will be moderating this conversation with Rebecca and asking questions on your behalf. If you have a question for Rebecca, please type it into the chat. If you have any problems with Zoom, we recommend exiting the meeting and rejoining us using the same link or meeting ID. If this is your first time using Zoom, you may need to adjust your settings and then restart the program. If you still have issues after restarting Zoom, please type into the chat and a staff member will assist you. And now I'll hand it over to Rebecca. Thanks for joining us.
Yes, thanks for having me. Okay, so before we get started, just a little bit about the way I've planned this. So I'll present a couple slides on some frequently asked questions. And it's really better if it's an interactive presentation and you're using the chat to send over any types of specific questions that you have that I haven't addressed. So please feel free to actively use that chat and send Amy questions during the presentation. And I'll take some breaks to answer some of those questions kind of throughout as I'm switching between topics. So to start, I'll just give a brief introduction to myself, like Amy previously said. So I'm a biomedical engineer. I just graduated last spring from Georgia Institute of Technology, and now I'm working on my PhD at Northwestern. So I've done some internships in different research and design fields with companies like Johnson & Johnson and Becton Dickinson. So not only am I familiar with how research works in an academic setting at universities, but I also am pretty familiar with industrial research. So what my goal is, as we increasingly hear about research on COVID-19, that term can be kind of daunting for some people who have never actually performed research or seen what a lab is like. So my goal is to answer any questions about how research happens and really facilitate the understanding of how we're trying to learn about this virus and mitigate a lot of the negative effects of it. So I'd like to start by talking about cloth face coverings. So the main reason that we're wearing these is to prevent transmission from asymptomatic individuals. What really separates COVID-19 from other types of viruses like the flu is that about 40% of people who are infected actually don't have any symptoms, or they're asymptomatic, which means that they can spread the virus to people who might have very serious symptoms without even knowing. And overall, this makes the spread of the virus pretty difficult to control. So one thing to keep in mind with face coverings is if you already have trouble breathing, they can be quite difficult for you to wear. So try not to wear them in long spans of time if you already have a pre-existing condition like COPD or asthma, and maybe schedule time throughout your day to take a break and breathe if you are in a situation where you need to wear these masks for extended periods of time. So also, if you're unable to remove the mask without assistance, it's not safe for you to wear. So keep that in mind when you're considering making public runs, because they are now required in all public places. Now, since they're required, it might be difficult if you don't have any sewing experience to actually acquire a mask. So what I've shown here on the slide is a way to fabricate a mask from a t-shirt alone, and it actually is a no-sew required technique. And if you're interested in using this technique to make a face mask, you can visit the CDC's website. They have pretty detailed instructions, and they're quite thorough on how to actually fabricate the mask from the t-shirt. One thing to keep in mind is make sure that your mask is 2 layers of fabric or double layered. so that it effectively stops the transmission from your mouth or your nose. So in addition to a t-shirt, you can also fabricate your own mask from a bandana. And this doesn't even actually require scissors. So if that's not something you have available and the t-shirt method wouldn't work for you, maybe this would work a little bit better. All you need is the bandana and hair ties or rubber bands, and you can craft yourself a mask as this diagram shows. And again, if you're interested, please feel free to visit the CDC website with the link that's at the bottom of the slide. Now, one thing to consider is face masks are really now part of our clothing. So don't think of it as necessarily something external, because what's happening is we have to wear these every single day. So now it's just part of our attire. So please remember to wash your face mask regularly. If you think about it, you're breathing into this for extended periods of time or even for short periods of time. And so it's really important to wash them. So in addition to that, this mask is helping to protect us from other people and helping to maybe protect you as well from maybe touching your face or touching your mouth or nose. So it's really important to wash these. Every day is probably best, but maybe if you're not wearing it every day, every other day or once a week might be okay. So in addition to that, if your face mask is particularly uncomfortable, if you're experiencing glasses fogging up, this might be a sign that the mask doesn't actually fit your face the right way. So like I said, a face mask is now like a piece of clothing. And we all have different sizes and structures to our face, which aren't necessarily being incorporated into face masks. So because face masks are so new, you can't necessarily go to the store and buy a size 5 or whatever the dimensions of your face might be. So it might be best for you to just try out multiple configurations. So if you're doing something like a t-shirt or a bandana, it might be easier for you to kind of manipulate the tightness of the strings or the way the fabric is folded in order to understand what stops your glasses from fogging up or what's the easiest for you to breathe while it's on. You don't want to be breathing and have air coming out the sides or the front. So if your glasses are fogging up, that's not a good sign, and it's not something you should really deal with. Also, you need to be able to see, so that's not something that you want. So the best advice that I have is to try out multiple fits if that's feasible for you. So moving on, I want to talk about how patients are tested for COVID-19. Testing is something we've heard a lot about because there's a shortage and of testing, and moving forward, we want to be able to test as many people as possible since, you know, we have that very daunting 40% are asymptomatic statistic. So the way tests are currently conducted is through a molecular biology technique that's quite complicated called reverse transcription quantitative polymerase chain reaction, or RT-qPCR. It's also just called PCR, which you might have heard before, and this right now is really the only effective, one of the only effective ways and the most widespread way that we have to test people. The way that this differs from something like the flu is that with the flu, the virus is very well characterized and we know what types of proteins that virus might express. And so we can characterize those proteins from substances like your saliva to identify whether the flu virus is present or not. COVID is different because it's not very well characterized. So the best way that we have is to directly try and identify if the virus DNA is within your saliva. So the way PCR works is, if you look at this diagram here, you can think of the pink blocks, the pink squares on the slide, as a piece of the virus DNA or a Lego block. And what we're trying to do during PCR is identify a location that piece of virus DNA fits within our own. So what we're doing is searching the patient's DNA for sequences that match that of the virus. So in reality, this test has a lot of different chemical components and a lot of different reagents that are required to make this reaction happen in order for that Lego block to find the sequence of DNA if it's there. In addition to all those chemicals that are required, there's also specific machines that are required. And the reason we hear about testing ramp up maybe being so slow or taking a lot of time is because not a lot of labs were equipped to perform these tests, because like I said, for viruses like the flu, there might be, there's other methods of characterizing them that labs are more equipped for. It's much less common that labs actually need to perform PCR in order to identify whether someone is sick with this virus. So if there's any questions, I can take some here. Otherwise, I will keep on going.
Yeah, we do have some questions. How reliable are test results?
That's a really good question. So in terms of false positives, really, it's very uncommon, because that Lego block that's looking for the virus DNA isn't going to bind unless the sequence is there. In terms of false negatives, because this assay is quite sensitive, that's more possible. So if there's an error in sample collection where the DNA is damaged, that could cause the Lego block not being able to bind the right way, and then it won't pick up the signal from the virus DNA like it's supposed to. So false positive is very unlikely, but a false negative could happen.
Okay, and so can you explain a little bit about why we have rapid testing and we have tests that take 2 days to get the results back? Why isn't it more consistent with how quickly you can get results?
Yeah, I think it's not more consistent just because different clinical spaces have different capabilities. So keep in mind that there are a lot of different components and specific site requirements to perform these tests. So the quickness of the results is really dependent on the processing and the time it takes for this reaction to happen. So I think in most cases, the slower results might be just the type of equipment that's used, because a lot of the variation between the tests that we have right now is just looking at different sequences of viral DNA. So this is really the primary approach. I think that some of the some testing sites might have optimized the logistics a little bit better, and that's why there's faster testing in terms of spreading the results and getting the results back to you faster. But I think it's really site dependent just on their internal capabilities.
So last time we had this discussion, we were talking about the lack of widespread testing, and at least in Illinois, we've seen a really, really kind of dramatic increase in the number of tests that are being conducted. Do you feel like it's enough? Do you feel like everybody should get tested? What's the threshold there?
Yeah, that's a really good question, and I'll talk more about that later as we think about starting to reopen the state and who exactly should get tested. But ultimately, it should be most people are able to get tested. So we will need to keep ramping up the testing, and that's part of the requirements of Illinois' now plan to reopen, is we can only reopen as fast as we have testing capabilities. Because so many people are asymptomatic, we need to make sure that we're identifying those people who are unfortunately asymptomatic and do have the virus, so that we can stop the spread and prevent maybe going backwards and having to re-implement shelter in place.
Got it. Okay. Going back to face masks, we're seeing all kinds of fashionable face masks, and you can get your favorite sports team on your mask now. And, you gave us some great examples of how you can make your own mask, even if you're not very crafty. Do those masks really work? And do they work as well as like medical grade masks?
Yeah, so medical grade masks are a little bit different because if you think about a hospital or a clinician who's treating a patient that has COVID, they are going to be in contact with the virus at a lot higher concentration and a lot closer proximities than we are in the public. In a hospital setting, a clinician could be going to patient after patient after patient that is actually infected and still trying to keep themselves safe from breathing in those particles. So those masks, the medical grade masks, are a lot higher duty than the cloth face masks. The aim of the cloth face masks is to just limit the spread, and they are effective in doing that between people in the general public, because the viral load in the air that we breathe when maybe we just go to the grocery store is a lot lower. Okay, so I think I'll keep going then. So now I'm going to talk a little bit about social distancing. So why exactly are we social distancing? So I'm going to start by introducing this concept of a reproductive number, and what this describes is how many people on average one person with the virus will infect. And this R0 basically describes how fast the virus will transmit and how intense we need to have social distancing. So R0 is actually reduced by social distancing, and that's why we have implemented this. One thing to keep in mind is that social distancing is actually separate from shelter in place. So shelter in place is more like the lockdown, where social distancing just refers to when we are in public, maintaining that 6 feet of distance, and also limiting physical contact and limiting crowds and large gatherings so that we slow the transmission that would normally take place in public places. So when we think about social distancing, why exactly is this something that's important? Thinking about China as a model is really useful because where the virus broke out, they really didn't have much of a notice as to what was happening, and they couldn't necessarily prepare and implement such a quick lockdown like we were able to. So if we look at Wuhan, where the virus started, there was about 50,000 cases with over a 4% fatality rate. In contrast, in the rest of China, there was only 13,000 cases with a 0.84% fatality rate. Now, what this shows us is when the healthcare system was overwhelmed, they were less able to treat the patients that had the virus, and that ultimately led to a higher death rate. But not only was the death rate higher, more people actually had the virus. So in the rest of China, where they were able to implement social distancing, the spread of the virus was significantly lower, as was the fatality rate. So this is kind of the justification and why we need to ensure that our medical system doesn't get overwhelmed, because it's a lot easier for them to actually save lives when they have the resources that they need and they're not overwhelmed. So if we look at this graph, just another way of maybe graphically showing what I just said, the red line here shows the spread of the virus when if there's no social distancing or no preventative measures. You can see that it has a very high spread rate, but it happens maybe over a shorter period than if we look at the green curve, which implemented social distancing, and there were overall less cases, but maybe over a longer time period. So this is really important, and this is kind of the type of modeling that public health officials were participating in when they were trying to decide what type of regulations to place on the public. But then also, if you see the blue line, there is a resurgence of cases when we stop the social distancing too soon, or before maybe the curve had all the way flattened. And what happens is there's a resurgence of cases. And right now, that's what we're really trying to prevent. But there's still a lot that we don't understand, so it is possible that unfortunately, we might have to go backwards if the virus begins to get out of hand and our healthcare system becomes overwhelmed. But in the long term, social distancing, where crowds are limited and we are remaining a safer distance in public, is going to be a long-term factor. Keeping in mind that social distancing is something that's separate from shelter in place. So thinking about Illinois reopening, we keep hearing about flatten the curve and peaks of the curve. So I wanted to show this data, and this is actually Illinois' basically curve. So what does the change in caseload look like all the way up to May 25th? And you can see that it appears that we peaked maybe about 2 weeks ago, and now we're on the down, the flattening, the end of the curve. So we can begin to reopen. As we all know, the shelter in place order is expiring, and this is part of how they make that decision. But how exactly are we going to get back to normal life and start actually reopening the state till we get to a point to where maybe we don't actually have to social distance anymore and we can start going to things like sporting events and concerts again? So here is Illinois' 5-phase plan for reopening, where phase 1 was kind of the initial shutting down of restaurants, the initial order for the shelter in place as the curve is rising. Phase 2 is the curve flattening, where you can kind of start to reopen things for curbside pickup and delivery of food or other maybe essential items. Phase 3 is when non-essential businesses start to reopen and we can actually start to have smaller social gatherings again, 10 or less people. And then we get into the later phases. So phase 4 is we still have to maintain cloth face coverings and social distancing. So this shows you that, as I mentioned, the face coverings and social distancing are really a long-term thing. And phase 5 is where basically everything is back to normal, or as normal as we will be, as if none of this ever happened, and we can start going to sporting events and concerts again. So technically right now we are in phase 2, but as soon as the stay in place order expires, we'll actually be moving into phase 3. So what exactly does it mean as we make this transition from phase 2 to phase 3, and how can we keep moving forward? So these are the metrics that Illinois has set for phase 3. We need to have less than a 20% positivity rate. So this means of all the people being tested, less than 20% have to actually be positive for COVID. And this informs us not only about the number of people who are testing positive, but it also informs us about the number of tests that we're executing. So as I talked about testing earlier, and we need to be testing a lot of people, as many as possible, the more people we test, the probability that this positivity rate stays lower. So as I mentioned, the positivity rate tells us about not only the transmission of the disease, but how are our testing capabilities keeping up with that transmission, and that's really what we're looking for. So as I mentioned, it is possible to unfortunately move backward in phases. So we can't have an increase of 10% over 14 days. So if maybe today we have a 10% positivity rate, and then 14 days from now we have a 21% positivity rate, that would be grounds for moving back or not moving forward in our phases. So in addition to the testing and the spread, we also have to make sure that our healthcare system is well equipped to actually address these cases in the instance that we do unfortunately have a resurgence of the virus. So Illinois is going to be working to maintain this health system readiness, and they're measuring that by making sure that we have no overall increase in hospital admissions for 28 days. In addition to that, at least 14% of ICU beds, medical beds, surgical beds, and ventilators need to be available. So if at any point the hospitals drop below that 14%, that's also going to be another warning flag. In addition, we have to have widespread testing for patients, healthcare workers, and at-risk populations. So those are the initial people that have been set aside, as we need to be able to make sure that we can test all of these people to ensure that no one is treating anyone if they are sick, and that's where healthcare workers come into play. We need to make sure that if a patient has it, we can tell when maybe it's passed, or we can diagnose them. And we also have to be able to test at-risk populations. And then the one thing that makes phase 3 a little bit different from the other phases that we were in, phase 2 and phase 1, is that we're going to begin contact tracing here. So I don't know if that's something maybe we've started to hear about in the news a little bit more, but what contact tracing means is that if I have the virus and I go to work and have close contact with my colleagues, and then I'm diagnosed with the virus, I have to be able to notify all the colleagues that I came in close contact with, or the state of Illinois will do it, that they have had a potential exposure. So the aim of this is to prevent asymptomatic transmission in the instance that I did actually pass the virus to one of my colleagues, but they're asymptomatic. So then they can begin self-quarantining and prevent spread as an asymptomatic carrier. Now, the way Illinois is planning to progress through this plan isn't the entire state will be on the same level. And that's just because considering we have the city of Chicago, there's a lot of different needs in Northeast Illinois as compared to maybe Southern or Central Illinois. So you can see here, the diagram is divided into 4 main regions, and each region will progress through the phases technically independently. And the way these regions were decided was basically based on historical public health data, and I think they also accounted for the initial trends in the virus spread when they made these regions. So I'm going to talk a little bit now about contact tracing, as I mentioned. So until a vaccine or a robust cure is available, the best way is to really identify people who may have the virus, and then have them self-quarantine until 14 days are up and they either remain asymptomatic or they have had a negative test. So this diagram here shows the way this might work. If person A is tested, so it kind of goes through the scenario I just said, where a person rides the train and then they go to work and then they go back home, and then the next day they get a positive test. Then the aim of contact tracing is to notify the other people they had close contact with. And the idea is that testing will be so widespread that all the people they had close contact with will be able to be tested. And meanwhile, the other people that they might not have had such close contact with, they can be advised that they might need to social distance or that they've had potential exposure. So I can pause here and take any questions if anyone has them at this time.
And I'm back. Sorry, I had a little issue with Zoom there. Just want to remind everybody, if you do have questions, just type them into the chat and I'd be happy to ask them on your behalf. Rebecca, I have a personal question for you. What are you most looking forward to when we move into phases 4 and 5 of this reopening plan?
I think for me, it would be nice to have like restaurants widely open again. I'm also a big White Sox fan, so it would be nice if we could go to sporting events. I don't know if that's like in the near future, but that's just me. I know that restaurants, there's a lot of talk about how they're going to reopen, but I think it'll be nice to maybe also just go outside without a face mask on. I think that would be pretty exciting to be able to do again.
Yeah, I think this stay-at-home order has been especially hard on sports fans because there's just, you know, if that's what you like to watch, there's nothing to do. I think that's been really hard. I'm not a big sports person, but I can definitely understand. I had a couple, if I can ask a couple more questions about masks. I got cut off previously. What do you feel about filters in masks?
Yeah, so I think filters and masks are a little bit unnecessary for the general public. Uh, they really should be saved for doctors and healthcare professionals, like I said, that are dealing with the virus at on a higher concentration, more widespread level, because they need to make sure that there are no viral particles that they are breathing in so that they can stay safe. For the general public, I don't think filters and masks are really necessary. In fact, they might actually impede your breathing more than the regular cloth masks. So just keep in mind that the cloth masks are mainly to protect other people if you're asymptomatic.
And when you are picking out your t-shirt that you're going to make your mask out of, is there a material you want to go for? I mean, do you want to stick with like a regular cotton t-shirt? You want to go for a poly blend? Is there some material that's better than others?
Yeah, so right now there hasn't been any guidelines from the CDC or other science saying that, cotton is better than polyester or anything like that. One thing to be sure is that you have a double layer of fabric. And also, if you're curious, the idea of the mask is to stop, particles from your mouth or your nose from traveling outside. I've seen some videos where people will actually take a spray bottle and just spray water through the mask, and then you can record it with your phone and replay it with slow motion, or maybe just watch it live and see how the water transmits through the mask. And the better the mask stops the transmission of the water or other particles like those that are coming from your mouth, the better it's going to work for you.
Okay. And then talking about contact tracing, if there is somebody in a workplace who is asymptomatic and test positive, what are employers supposed to do with that information? Is there more of a risk of transmission from an asymptomatic person versus a symptomatic person? Are there recommendations on when to shut your business down? What's the plan there?
Yeah, that's a really good question, and I think it's going to be up to the individual employer. Right now, it's the state of Illinois, it's the public health department. It's their responsibility. I don't know if that's the right word, but it's Illinois Department of Public Health is really the ones leading the contact tracing. So I don't know how much your employer is necessarily going to be involved if there is someone in the workplace. It could be possible that we see regulations from the state of Illinois saying if this many people have come in contact that, you have to take this extra precaution. I'm really not sure how Illinois Department of Public Health is going to handle that with the individual employers. Maybe we'll see more guidance when we actually start phase 3, or maybe they'll disseminate that information privately. In terms of symptomatic to asymptomatic transmission, I think theoretically there's no difference in transmission between those two types of people, between asymptomatic or symptomatic individuals. On one hand, an asymptomatic person might be more inclined to think that they don't have the virus, and potentially there's a risk then for them not social distancing as rigorously as they need to, or not realizing that they need to self-quarantine. So I think in that respect, asymptomatic individuals might be a little bit more risky compared to the symptomatic ones, since the symptomatic people are likely more inclined to take a COVID-19 test and get tested, and then they'll understand for sure if they need to self-quarantine, where an asymptomatic person might not actually take that step because they don't realize that they're sick.
Great, that's all we have for right now.
Awesome. So thinking forward, we know we're geared up to go into phase 3. So I put these images here. These I got actually from the Illinois Department of Public Health website, and these refer specifically to the Northeast region of Illinois, which is what we are. And you can see, if this is something that you're curious about, exactly how are we hitting these metrics? How are our hospital resources doing? And this is really a good resource. It's quite transparent, the website, if you're interested in seeing how close are we at moving to the next phase, and is there a risk of us moving backwards? So these numbers I actually got today, so they're updated. And right now we have a 14.5% positivity rate. And remember, the metric for that for phase 3 is less than 20%. And you can see how that positivity rate has changed in the last 14 days, and we're actually going down, which is really promising. There's been a decrease in 4% of positive cases. And in addition to that, there's been a 50% decrease in hospital admissions since May 1st. And then looking at the lower level of these green graphs, you can see that our medical, surgical bed, ICU bed, and ventilator availability is quite high. So that has to stay above 14% in order for the state to remain on the path forward. And you can see that our hospital resources, in terms of resources that are available to the patient in the case that you do contract COVID-19, is quite good right now. So on the right, you can also see the graphs over 7 days. What you see here is an average that's plotted, and our medical and surgical and ICU bed has gone up, and our percent positivity tests are still going down, and that's over an average of 7 days. So it really looks like right now we are on the right track for ending the shelter in place and reopening. But we still want to think about moving forward, moving into phase 4, where basically a lot of things are reopened, but we're still social distancing. We're still wearing face masks, but you can start to have larger gatherings of less than 50 people. So at first glance, when you look at this, these metrics, they're all very similar to that of phase 3. The two that stand out that make the jump is widespread testing regardless of symptoms and risk factors. So if you remember, for phase 3, we needed to satisfy widespread testing for healthcare professionals, at-risk populations, and patients. So with this, we'll have to make the jump to more widespread testing regardless of if you have symptoms and risk factors, so that we can identify the asymptomatic people with more efficacy. So in addition to that, the contact tracing also changes in that the goal is to have contact tracing for 90% of cases in the region. So that seems quite steep at first. So what that means is that the state wants to be able to contact 90% of the people that an infected person might come in contact with, to let them know that they're at risk. So the idea is that this will be possible because as we increasingly see a decline in cases, that it'll be more manageable to reach out to these people who have potentially been in contact. So, and in order to do that, in order to also effectively maintain this contact and have people identified if they have close contact, we need to have more widespread testing so that we can test people even if they're asymptomatic. So those are really the key indicators of phase 4, in addition to maintaining the less than 20% positivity rate and hospital resources that we previously talked about. So in addition to that, what we're really aiming for is really a vaccine, so that everyone can be protected and we can develop herd immunity. But the issue with that is that vaccines take a really long time to develop. So briefly, I'm just going to talk about the different phases of vaccine development and why maybe vaccines take so long. Under normal conditions, vaccines might take actually up to 10 years to develop. So the first step is to think about some sort of formulation that might work, or some sort of biological strategy, and apply that within a lab. So that might mean to cells, or it might also mean to an animal model, and then basically making sure that the theoretical treatment is safe and it's also effective. So once a company or a university has something that they think is going to work as a vaccine, then they can submit to the FDA and try to begin clinical trials. So there are 3 phases of clinical trials, and these involve humans. So that means that each of these phases also requires a human volunteer to receive the experimental treatment. So this actually takes a really long time. Some recruiting can take normally up to a year to actually get people to volunteer to be tests for, this experimental chemical concoction. And even once all that's done, manufacturing can be pretty complicated, and it's often something we don't think about when we think about developing a vaccine. But these biological molecules that are being packaged are quite sensitive, and we want to make sure that they stay safe when they're packaged and that they also will work after being on the shelf for a certain amount of time. So this is just a little look at what the landscape currently looks like right now and who is trying to develop vaccines and how many are there. So you can see that there's actually quite a lot going on, primarily by private companies like Johnson & Johnson. And in addition to that, these vaccines are in a lot of different stages. So the graph on the right, the exploratory stages, that's what I refer to as kind of in-lab development. Something that you could also think about is, in order to test in animals, researchers also had to develop a COVID animal model, which is not something that we had. So that was another hurdle for this vaccine development. But exploratory and preclinical basically refers to lab work, basically before submitting to the FDA. And then only 5 companies right now are currently in phase 1 clinical trials. And I think they are all in the recruiting stage. So they're all trying to get human volunteers to actually commit to trying their vaccines. So in addition to all these different places and all these different stages of vaccine development that we're currently in right now, you can also see at the bottom right corner, there's a lot of different strategies that are being involved in the vaccine development. Some of these strategies haven't actually been attempted before for vaccines. So there's a lot of different innovation that's going on right now to try and solve the COVID problem. So these are the 5 vaccines that, as I mentioned, are in preclinical or are moving into phase 1 of clinical trials. I'm not going to talk about the specifics. What I want to show here is that each of these numbers you can actually use to track the progress of the clinical trials. So the NIH has a website called clinicaltrials.gov where you can look at the vaccines and each of their status. So this is one example that I showed, that I pulled from the website, and anyone can access this. And it tells you the name of the company, but it also tells you their approach to the vaccine, what it is they're using, when they started, and when they anticipate their clinical trials to be completed. So you can click on the results tab, although right now there's no results, and it will tell you what phase the vaccine is in of its clinical trial. So you can see here that the specific vaccine that I clicked on from that table that I looked up with that number is in the recruiting stage. So they're still trying to get patients to commit, or volunteers to commit to their study. And they estimate completion around April 2021. So that might seem far away, but it was actually one of the closest times for the vaccine so far. If you're interested in looking at other COVID-related clinical trials, you can also go to their website, and the link on the top of the page, at the top of the bar, you can click on that and it will take you specifically to all the COVID-related clinical trials that are being executed right now for vaccines, as well as drug development. So if that's something you're interested, or you're interested in maybe seeing exactly when the vaccine is coming, this is quite transparent and not usually something that you have available to you. A lot of companies don't disclose their clinical trials, but I think because of the nature of COVID, it's becoming a lot more easy to try and track when exactly the vaccine is estimated to be finished. So I think I'll take any questions here if there are any.
So there's been a lot of talk about, when we can reopen, and that seems to really hinge on when we'll have a vaccine, but there's also been talk about having a treatment. And I know that there's been some promising information coming out of some of the like remdesivir and some other treatments, but there's not really like a cure for this or a treatment other than kind of supportive care. Do you think that we'll ever have a vaccine or a treatment, or do you think that this is going to be something that we'll deal with kind of like the seasonal flu?
I think a vaccine is definitely possible. Vaccines for viruses, especially viruses like the flu, are pretty manageable. And there's been a lot of other vaccines for viruses that have been developed in the past. And as I showed, there's a lot of different companies that are currently making the push in such a short time. So I definitely think there will be a vaccine. In terms of a cure, viruses in general are very difficult to treat just because of the nature of how they infect our cells and then basically turn our cells into machines that produce what they need to survive. It's very difficult because our cells are the ones that are housing the viruses and allowing them to replicate. So that's why it's really difficult to find a cure a lot of the times, especially for the flu. The flu is a good example of a way to think about it, in that we can predict what the strain will look like each year and develop a vaccine. So in the case that COVID is like that, and it is a seasonal virus, I think it'll be pretty manageable to develop a yearly vaccine in a similar way that we develop a vaccine for the flu each year. Although that is something that we don't know, it is possible that you can just get it once and have long-term immunity. So there's a lot of things that I think we're still trying to figure out there.
That's pretty hopeful. And it's good to hear, because we're not hearing a lot of very hopeful things right now in the news and research and everything. Can you talk a little bit about the value in antibody testing? I know that there are some commercially available tests that you can get without a doctor's order. Do you think that there is a reason to have that test? Do you think that it is accurate enough? Can you talk a little bit about that?
Yeah, that's a good question. So antibodies are interesting because what they show when you get the test, it shows you if your body has had to fight the virus and has learned how to produce antibodies in the past. It's good to get tested because potentially you can become an antibody donor and donate antibodies to someone who might need them, who has the virus. There's been some evidence that the antibodies are helpful in preventing someone who, like a healthcare worker, from getting the virus until we have a vaccine. The goal of a vaccine is to encourage your body to produce antibodies that it would naturally produce if you were sick and had the virus. So maybe donating those antibodies is a beneficial thing because they could actually help someone who has the virus, since we don't have a really solid cure right now, but they can also help healthcare professionals until we have a vaccine. In addition to that, in terms of, if you do have the antibodies and you've found that you did have the virus and you didn't even realize it, it might be helpful because you can realize, the asymptomatic transmission, but that doesn't mean that you are necessarily not going to get the virus again, just because we don't know how immunity works. So it might be risky if you see that you have the antibodies and then you can think that, you're safe and you don't need to social distance, because that might not be the case. And in that scenario, if you contract the virus a second time and you're asymptomatic again, you risk giving other people the virus who might be at a higher risk and not asymptomatic like you were. So I think just take your antibody tests with a grain of salt and keep in mind that it doesn't mean that you're immune. In terms of the accuracy of the tests, I've also seen that what has happened is there was a big push to get antibody testing released, and maybe the tests weren't as rigorous or as accurate as they should have been. But we're working on, I've seen a lot of research that's working towards refining those tests and really making faster and more accurate antibody tests available so that we can have a better way to actually get those results.
Okay, and then we have one last question before we move on. It's more of a comment that has turned into a question. The comment is about how some of this statistical data that's coming out in various press conferences and from the State Department and Chicago Department of Public Health is really confusing. And I agree. I mean, I have a background in data science, and sometimes it's difficult to see it without putting it in context. But the question is there a way to compare, this time period's overall death rate, and we're tracking deaths due to COVID. Is there a way to compare this time period versus the previous year's time period? Is there a way to kind of tell if, the overall death rate is increasing? And would that be a better way to tell, kind of the I guess, morbidity of this disease?
Yeah, I think it's pretty difficult to determine at this point because there's a lot of factors that go into play. I think that the diagram about what happened in China is really powerful because it shows just how unpredictable the death rate of this disease can be. Because you look at Wuhan and there's about a 4.5% death rate. But then you look at the rest of China and there was, about a 1% death rate. So it's really difficult to compare because the biggest problem with this disease is the transmission. And if we didn't lock down the transmission with the measures that we're taking, the death rate would be a lot higher. So it's hard to say how deadly is the virus when we're doing so much to make it less. A lot of the actions that we're taking are really reducing the transmission of the virus, not the virus's deadliness. And I think that's something that does get lost when we look at statistics, is we can say, it's easy to say the virus isn't killing that many people, but it's really just not as many people are getting the virus that the virus could be deadly to. Because we have such a good understanding of asymptomatic transmission, 40% is an incredibly high number, which is not something that we see with the flu. Typically, people who are asymptomatic with the flu can't transmit it. So that's why it's really difficult to compare these two numbers, because there's so much that we don't know about how this virus acts. And it is possible that maybe we have short-term immunity and then the virus changes and becomes less deadly. So in terms of tracking the death rate of the virus that way, that might be something really helpful, but I don't know that there's a solid way that we can compare on the same level. We're really comparing two different things just because of how we're handling this virus, more with stopping the transmission than actually reducing the deadliness of the virus.
Great, thank you. I think we are ready to move on.
Okay, so I wanted to just talk about managing stress, since we know that mental health is really important and it's linked to your immunity. So One of the biggest things that I've seen is to take breaks from the news each day. If you schedule time to take out from your day, maybe just this is 1 hour that I turn the TV off, it can be really good to just take a break from that because it can become overwhelming. In addition to that, try to keep your body healthy. As the weather gets nicer, it becomes a lot easier to maybe take a walk outside or plan time for other types of events that you enjoy every day. And with that being said, make sure you're checking in on your friends and family to make sure that they're all doing all right, just because, you know, you have to be careful about staying separate. That doesn't mean that, you know, you can't find other ways to do things together that are safe for everyone and maintain social distancing so that you can feel more connected and really keep your morale up. So I wanted to post here about some informative resources. So number one is medical professionals. Listen to your doctor in the case that you need testing or unfortunately have the virus. They will direct you with how to handle it within your household and what measures that you need to take. There are also open source research journals. So if you find any of the things here particularly interesting, you can take some time to search into those things independently. In addition to that, company websites are actually quite transparent about some of their drug, vaccine, or medical device development, just because this is such a widespread issue right now. So some of their websites can actually be quite helpful. I've linked to Johnson & Johnson here just because they have a video series going on called A Path to the Vaccine. That can be interesting if that's something you're interested in. In addition, the Illinois Department of Public Health posts statistics regularly. So if you want to see how we're doing in our phase 3, when can we move to phase 4 or 5, you can go there and see the graphs that I posted here with like the meter that shows the percent change and what phase we're going to be in. The CDC website is also really good. They have a lot of general types of resources like the masks that I had shown here. And then the library. Amy, if you want to say a couple words, I know that the Glen Ellyn Library is doing a lot right now. If you have any questions.
Yeah, I just want to take this opportunity to remind you that the library is your public information resource. And, you know, this is what we do. It's our bread and butter. We love talking about helping you find reliable sources. If you're having trouble understanding, you know, statistics, we can help you with that. If you just have a question, you can give us a call and we love to answer your questions. I know that that's true of Orland Park as well. I don't know how many of our participants are from which community, but I just want to remind everybody that you can contact your library. We are ready for your questions. We love to handle questions. And even if we don't know an answer, we know how to find out an answer. And we're, you know, in constant communication with people like Rebecca and other community members who help us understand this information.
Awesome. So I'll take any more questions if you guys have any. I've put my email here in case you're interested in any of the resources I posted. If you're interested in looking more into the clinical trials or the vaccine progress and you're not sure how to do that, please feel free to email me. And thank you guys for coming today.
Thank you, Rebecca. I don't see any other questions in the chat, but if you do have questions, type them in. We've got a few more minutes before we end for the day. I have a question about donating blood. So we've been having blood drives at the library in the parking lot in a bus. And I'm just wondering, is it safe to donate blood? You know, it seems like we're always kind of on the verge of a blood shortage. What are some things to consider if you're thinking about donating blood, and especially what are some reasons to do it now more than ever?
Yeah, so right now donating blood is really important because there are shortages since people were kind of afraid to go out and maybe interact with medical professionals in settings that they felt were particularly risky. But donating blood is very safe, and it's very important for treating maybe other ailments besides COVID. There's a lot of things that blood can be used for, and they still need it, but there's a shortage, unfortunately, just because of social distancing and shelter in place. So it is definitely safe. And, you know, if you get antibody tested, it's something that you might want to think about, because there are a lot of really, really good things that those antibodies could be used for. And they can use your plasma and maybe treat someone or send it to healthcare professionals so they can kind of use the antibodies as a vaccine while we are still working on one.
Okay, and do we know for sure yet how long the virus lives on different types of surfaces and how long it lingers in the air?
Yes, so that's definitely something that we are still trying to figure out. In terms of lingering in the air, your virus, the virus is attached to, you know, like particles when you speak or breathe. So it only lingers as long as the particle that it's traveling on lingers. We're not sure exactly how long it lives on different surfaces, but we do know that these surfaces can be feasibly disinfected, and then the virus doesn't live through disinfecting. So as long as you use proper cleaning agents, it's definitely you'll be safer that way. But we're not sure yet how long it lives on different types of surfaces.
I think everybody is anxious to find that out too. Can you talk a little bit about health inequity? You showed us a diagram of China, and we know that, you know, highly populated, you know, urban density, you know, really is a risk factor here, because it's so much harder to, you know, social distance when literally walking out your front door puts you in close proximity with others. But in places like Chicago, which doesn't have a completely uniform population density when you compare different parts of the city, we have, you know, pockets of, you know, higher infection rates and higher death rates. And I'm just wondering if you can talk about some health inequity and why that might be a factor in that.
Yeah, so some of the biggest things are access to information. So I saw a study that showed when the virus outbreak first started, people from lower maybe socioeconomic statuses didn't really understand the virus, especially when you have a lot of conflicting sources of information that are saying different things about how deadly the virus is. That can maybe confound things in terms of the seriousness of social distancing. So I've read that was one major reason for maybe higher death rates in certain places of Chicago, was just the access to information. But then also you have to think about different education levels. So maybe there are less college graduates or less high school graduates in those areas that can understand what types of information are the most credible, or how to interpret that information. So I think all these things go into play when it's really harder to receive information and you have less informative sources, but at the same time, it's harder to interpret the information that you are getting from all these different sources. You can think about, you know, maybe if the only news that you have is from Facebook, you might think about things differently than if the only news that you have is from maybe ABC, or watching the governor's press conferences every day. So I think that the availability of information is the biggest thing there.
Yeah, I think there's also a growing understanding of the racial disparity and health equity. You know, we've got parts of the city that have a poor public health infrastructure, and you know, I think that's kind of like coronavirus and COVID-19 are showing, kind of like shining a light on that right now, because we've always kind of been aware of it, but now we're seeing it in action. Are there any lessons that we can draw from the 1918-1919 Spanish flu pandemic and reopening, and, you know, how it was handled 100 years ago, that may kind of help us understand this pandemic?
Yeah, that's a good question. I think one thing to keep in mind is everything is very different now than it was then. So there are some there is information we can take, but it all has to be taken with a grain of salt. One thing that we've seen, and it has already informed a lot of public health response to COVID, is that cities who quarantined and social distanced more intensely during the Spanish flu had better economic outcomes after the outbreak had passed. So that had informed a lot of the state response to COVID and how we social distanced, because you have that thought of seeing what the after effect was in different cities in the Spanish flu that had or hadn't quarantined. At the same time, we have technology now that's a lot different. So we also have the ability to work remotely a lot more feasibly than they did in 1918. So is that economic, benefit going to transfer over as directly? It's really hard to say at this point. And we also have a lot of enhanced technology. So we were quite aware of the situation before it maybe got here, because, like I said, information spreads a lot more rapidly now. We have a lot more access to different sources than they probably had in 1918. So were we more prepared to start? Potentially, and maybe that also mitigated some of the deaths, because we understood a lot more about the virus. Even though it seems like there's still so much we don't know, we understand a lot more than they understood about the Spanish influenza in 1918. So it's hard to draw comparisons, but I think it does, and it has been used to inform public health policy.
Got it. And one last question before we adjourn for the day. If our attendees for this presentation remember just one thing from today, what would you like that to be?
Yeah, I think the biggest thing is realizing that there is a lot of really good progress that's being made. And although it can seem overwhelming with all the information that we still don't know about the virus, that there's a lot of positive decisions that are being made through science to really drive vaccine development and ensure that we're social distancing and stopping the transmission, and really doing the best that we can to mitigate the virus. And it's really a lot of baby steps, but there's still a lot of positive things that we're learning about science from this virus.